My master's study was based on the implementation of molecular methods for the characterization and identification of pathogenic E. coli for routine and industrial application. As we all know, E. coli is poo bacteria, which ends up in sewage water, goes through a purification system and eventually ends up in environmental waters. Or it may end up directly in environmental waters due to human activity. This poses a huge problem to people using it because it may cause disease. Why? Because E. coli contains pathogenic strains, and these pathogenic strains are known to have caused diseases worldwide. Also, testing for pathogenic strains of E. coli may give us an indication to the different pathogens that may be present. This study used molecular methods on environmental water samples. We sampled water from the final effluent, upstream of the final effluent release point, and downstream of the final effluent release point. All of these water samples were subjected to DNA extractions on a 96-well extraction method. This method was an automated system which we adapted onto a semi-automated system. The resultant DNA was then used in 11-gene multiplex PCR. The results from the multiplex PCR enabled us to characterize the different E. coli into the various pathotypes. During the study, we found that the final effluent did in fact have pathogenic E. coli strains. We also found that the upstream and the downstream points did have pathogens before the release of the final effluent. The 11-gene multiplex PCR also enabled us to find combinations of genes. Gene combinations which enabled us to characterize the E. coli into the different pathotypes, as well as combinations that were not specific to any pathotype. This gives us insight and opens up avenues for us to study single isolates, where we will now characterize the different gene combinations. Also, we need to study antibiotic resistance, because as we are now finding that these bacteria are becoming more and more resistant to antibiotics. All of this work will now form part of my PhD studies. All of this is important because if we do not understand it, we cannot do anything about it.
Please hang up and try again.